This video will introduce Chapter 32, which is just an overview of animal diversity. We're going to be looking at some of the systems and characteristics that we'll be seeing in our animal phyla. So when I say animal, most people think of the big, charismatic, fuzzy animals, usually vertebrates. So we're going to see later that most animals are actually invertebrates, things that lack a backbone. So all animals are multicellular and eukaryotic. So this is one of our kingdoms within the domain eukarya. All animals are heterotrophic and they ingest their food. So this is different than our fungi, which absorb their nutrients. Animals do not have cell walls. And at some point in their life, they will all move. Okay. They also have the ability to reproduce sexually for most of them. They have tissues such as nervous system and muscles with a few exceptions. And they have specialized sensory structures often located on a head if they have one. So one way we get diversity within animal bodies is this Hox gene. And the Hox gene family has been highly conserved. So if we look at the actual sequence, it's the same over and over again in each species. But the repeats and the location of these repeats give us all types of body forms. So animals have a common ancestor with choanoflagellates. So this is a type of protist, and if we look at the shape of these choanoflagellates, it's very similar to what we see in certain animals, especially our sponges, the ancestral or first branch of the animal kingdom. So these choanoflagellates are well known for these little finger-like protrusions, this comb that surrounds the flagella. And there are collar cells in our sponges that look almost identical to those choanoflagellate protists. So animals are divided based on body plans. So we're going to look at some distinctive traits, things like symmetry, the presence or absence of tissues, and what kind of tissues they have, whether or not the organism has body cavities, or their patterns of embryonic development. There are three patterns of symmetry we're going to look at. The first is asymmetrical, or the lack of symmetry. So this sponge down here is very odd shaped and has no symmetry. The second is radial symmetry, and this is when we can take organisms and cut them on many planes and get pieces that look relatively the same. So a pot has radial symmetry, this anemone has radial symmetry. It's kind of like a pie or a pizza. Bilateral symmetry is our third example. This is when you can cut things down one single plane and get two parts that look relatively the same. Once you see bilateral symmetry evolve, organisms generally have a head, and we're going to see that there are different sides to the organism. So a top and a bottom, so dorsal and ventral. There's left and right, anterior, which is the front, posterior, which is the back. And because we now have these directions, we're also going to start seeing a change in perception. So these organisms are going through life going in one direction, so straight forward, leading with their head. And we also see the evolution of many types of sensory organs on that head. So the squirrel, for example, has eyes and a nose and two ears. So lots of senses located right there in the front so that they can perceive what's coming at them. If we look at radial organisms, they're not perceiving anything from front and back. They kind of take in their environment in a 360. So if you think about a jellyfish, for example, they have sensory organs all the way around their body. So they take in everything in a 360 all the way around, top and bottom. And if you watch a jellyfish swim, it will actually swim up, and then it will swim sideways, and then it'll swim upside down. So this is partially due to their radial symmetry and the lack of a head. The next way we categorize animals is based on their tissue. So some animals do not have tissues at all. Other animals have specific types of tissues. So a tissue is just a collection of specialized cells, and these cells are similar and they're working together towards a similar function. Okay, so if you think about a tissue, it would be something like your muscle. It's a bunch of the same types of cells working together to contract and cause motion. During development of embryos, we see that many animals have 3 germ layers that can give rise to different types of tissues. So when we look at tissue development, there are 3 types of tissue. The ectoderm is on the outside of the embryo surface, the endoderm is the innermost layer, and the mesoderm is the middle germ layer or middle layer of tissue. And the mesoderm is where we see a lot of tissues and organs develop. So some animals, like sponges, have no true tissue. Other animals have 2 layers of tissue. We say they are diploblastic, and they only have the ectoderm and the endoderm. The triploblastic organisms have all 3 of those tissue layers. So they have endoderm, ectoderm, and mesoderm. The next trend we're going to look at is body cavities. So most triploblastic organisms have a body cavity, but some of them are acoelomates. So A means no or non. So acoelomates have this outer layer, their ectoderm, inner layer, the endoderm, and then the mesoderm completely fills up the other space. So there is no room for organs or other things in here. The pseudocoelom is a false body cavity. So one side of the cavity is lined with the mesoderm, but it's not a true and completely separated chamber. Okay, so remember, blue is going to be our ectoderm, yellow is our endoderm, and then our mesoderm is just going to line one layer. So there's a partial body cavity here. Organisms with a true coelom, which are either coelomates or eucoelomates, have a completely lined body cavity. So this mesoderm is going to line the ectoderm, it's also going to line the endoderm, and it's going to create this body cavity that they can put other things in, like their organs, so hearts and stomachs. So having a body cavity is a trend towards increasing complexity. It provides extra space for extra complex structures. The next thing we're going to look at is embryonic development. So right after fertilization, we have a zygote formed, and this is a single cell. We then start to have cleavage or mitosis occur, and we see this ball of cells form. And that ball of cells is eventually going to hollow out into this blastula. Okay, that blastula will then start to pinch, and we have a gastrula forming. So we can categorize animals in 2 ways based on their embryonic development. Protostomes have spiral determinate cleavage. So if we look at the lines here, we see that they are not even, okay, and these cells are not sitting perfectly in line with each other. If we look at the deuterostomes, we see that the cleavage is radial and indeterminate. So we have lines going all the way down this little embryo. The next thing to look at is the blastopore. So this hole that pinches into the blastula. In protostomes, this blastopore is going to develop into the mouth of the organism. With our deuterostomes, that blastopore is going to become the anus. So protostomes, the mouth develops first, and in deuterostomes, the anus develops first. So remember, our simplest animals have no tissue. Eventually, we're going to see the development of tissues, and it's going to start with basic things, a nervous system, maybe some muscles. Eventually, that's going to get more advanced, and we're going to start seeing organs, which are groups of tissues that are specializing in a particular function. So your stomach is an organ. It's made up of many tissues working towards digesting your food. And then we're going to see organ systems. So bunches of organs working together towards a greater goal. So your digestive tract, for example, is a set of organs all focused on breaking down your food, and they each have a different part to play. So speaking of digestive tracts, we're going to see two types of digestive systems in these animals. The first one is an incomplete digestive tract. So this means that there is a single opening, so food will go into one opening, it gets digested, and then the waste materials are going to be expelled out that same opening. Okay, so it's kind of like a sac system. The other type of digestive system is a tube system. So you're going to have a mouth, so food is going to enter in one end, it's going to pass through the digestive system, and wastes will exit out the other end. Okay, when we look at circulatory systems, we're going to have open circulatory systems and closed circulatory systems. So an open circulatory system has a heart, and blood is going to be pumped from that heart out into open body cavities. Okay, to get the fluid back into the heart actually has little holes that accept the fluid back in. This is different than a closed circulatory system. So in closed systems we have a heart, there's a set of tubes that leave the heart, the blood's going to go out to the body tissues, and then it's going to return to the heart all in those vessels. Okay, so a closed system, blood is always contained. In an open circulatory system, blood or hemolymph is pumped into body cavities. And we're also going to see a variety of respiratory organs, things that allow or help with gas exchange. So in mammals, we see lungs. Birds also have lungs. Some organisms breathe through gills. Others use their skin for diffusion. So there's a wide variety of respiratory organs in the animal kingdom. Support systems also vary. So many of our simple animals rely on a hydrostatic skeleton. So if you think hydro, we're talking about water, and this is just using water pressure to maintain their shape. Some organisms are going to have an exoskeleton, so hard structures on the outside of their body that are used for muscle attachment and movement. And then many organisms have internal support. So you and I have an internal skeleton. So this slide has 5 important facts that we have learned about animals. So looking at the phylogenetics of these organisms, we see that animals all share a common ancestor. Okay, we also know that sponges are the basal group or the outgroup of our tree. We also know that eumetazoa, or true animals, all have true tissue. So there's only one group, the sponges, that lack true tissue. And most animals also belong to the clade, or the branch Bilateria. So this means that most of them have a head and bilateral symmetry. And there are 3 groups within this bilateral grouping, and many of them are invertebrates. So lacking a backbone. There's only one phylum that includes vertebrate animals. So Chordata talks about having a spinal cord, and we're actually going to see that some animals within this phylum do not have vertebrae, so there's actual bones to protect that spinal cord.